We seek to understand how cells organize themselves in 3 dimensions, how they reproduce themselves in 3 dimensions. We're all about space and time at the level of a single cell and below. We're focused on how the various portions of a cell are organized internally and how the cell relates to its neighbors.
I joined as part of the BBS program, the Biological and Biomedical Sciences.
Since I have the comparison between the department in Germany and the Yale School of Medicine, I think here the connection between people is more present.
Being part of this department has been great because of the access to so much great science. There are constantly talks in the department and across different departments too that we have access to.
We also work really close together, so either PhD or postdoc or technicians.
We take pride in the inclusive, collaborative atmosphere that we have in the department. Many, many collaborations exist. It's actually a rare project in one of our labs that doesn't involve another lab, either because of the subject matter or technology.
The whole environment of having not only the people but also the resources, which has been really amazing.
If you ever wanted to do any technique, there is always someone to go to. So it's really the limits of your imagination, what you want to do with your science.
The department was founded in the mid-1970s by George Palade, who had pioneered the use of electron microscopy, an imaging method. And as a result, we're the first to discover the principal components of the cell, ribosomes, membranes, organelles like mitochondria, endoplasmic reticulum, and so on. And actually, those series of very fundamental questions resulted in 2 Nobel Prizes in physiology or medicine that the department received, first to Dr. Palade and actually the second more recently to myself. And what separates cell biologists from the many other types of biologists we have in universities and in medical schools is the level at which we study biology.
We are working on membrane trafficking, and my group especially is working on protein sorting and secretion at the Golgi apparatus.
I work on energy production in brain cells.
The kinds of questions we ask are very fundamental. The fundamental aspects of life are conserved. They have broad impact, and they allow us to understand the basis of many human diseases, for example.
There are a lot of degenerative diseases where neurons are not able to provide the requirements to allow a functioning nervous system.
What happens when these enzymes or these proteins that produce energy in our brain cells, when they get disrupted, how that affects brain function?
And this is actually the very important part to find out how diseases developing and how to cure the disease, because when you know it, you can have an anchor where you can potentially develop some pharmaceuticals or medicine against this.
But as much as we have learned, there's so much more to learn. We've had major discoveries just in the last several years even that have opened entirely new vistas of cell biology.
There are a lot of things that are known, for example, all these organelles that can be seen by the microscope, but what actually happens inside the organelles, or what are the dynamics, or what we are doing trafficking, is not so much known.
We're at the beginning of another era of discovery in cell biology, and the Department of Cell Biology at Yale is equipping itself for that purpose.
This department specifically has a lot of state-of-the-art imaging technologies that allow you to see things in cells that have never been seen before. So I think that has the potential to lead to new discoveries.
If you have an interest in the fundamentals of life and you wish to have an impact on the world through that interest, I'd like you to consider our department as a place to come.